All right, cool. So really excited to give this presentation on some of my work in open source drug discovery and sort of what I've been doing with the help of the Emergent Ventures grant and some of the research I've been working on. So just to give a little bit of a basically intro, my name is Seon. I'm basically 17. I'm a high school senior. I'm from Toronto, Ontario. And basically my research is focusing on applying machine learning towards drug discovery and basically chemistry research. So trying to understand different features that cause molecules to bind to proteins or different drug targets. So basically some of the things I work on is I'm a research intern at the University of Toronto with Professor Alan Asperguzic. And I'm also on the developer team for DeepChem, which is an open source project that's applying machine learning towards the life sciences and drug discovery. So in the past, I worked a little bit on enterprise software. Actually, I worked at a company called Integrate AI, which was basically deploying these customer solution software for different basically consumer software companies. And yeah, so I guess I'll get started a little bit. So when you look at drug discovery today, drug discovery is pretty much this billion-dollar 10-year sort of pilgrimage. So you have to generate thousands of different drugs to be screened, which this process takes around 3 to 5 years. And after you get to the process of generating these huge assays of molecules, you start to do in vivo testing in the lab on different animals or maybe different cell cultures. And this takes another 1 to 2 years. And if you manage to get, say, your compound to that point, then you go through this huge clinical trial process. And that takes largely like 6 years because you're going through 4 clinical trials and these are human trials. So this is looking at like your standard small molecule drug discovery pipeline, which would be largely different than something like the COVID vaccine pipeline, which has largely actually accelerated this pipeline just because of sort of the new met need for this vaccine. And the drugs that manage to pass through all 4 of these clinical trials finally get to basically market and then get to regulatory approval. So this is a really large pipeline for pharmaceutical companies. So it takes up to $1 billion and it's over a decade long. And I think the really sad thing about this is that this sort of inhibits a lot of small biotech companies from, say, taking a drug from designing the drug to actually bringing it out to market. What happens to most drug discovery startups is once they generate some that, say, show some promise in the lab or in animals or in maybe the early clinical trials, they'll sell them to, say, the bigger pharmaceutical giants like Novartis or Pfizer. So it really sort of inhibits a lot of innovation in, I think, biotech largely. And I think that's something that's starting to change a little bit. And sort of the reason why I believe that's changing is really open source science. So what I mean by open source science and computation is that I think with open source science, you're basically allowing anyone with a computer to generate value in the discovery of new drugs. So using packages for chemistry, for biology, and for machine learning, you're able to, say, design a series of molecules, computationally test those molecules on a target of your choice. And all of this is relatively easy to do. There's lots of resources for learning how to do this, textbooks, courses, and even tutorials. And I think even the project I've been working on, DeepChem, has been trying to sort of make this process easier to people outside of academia and industry. And so sort of the goal with this is that by building tools which more people use, you sort of increase the chance for the discovery of new medicines, sort of when you're playing this whole lottery that is drug discovery. So just to take a step back and sort of tell you how I sort of got here. Before sort of high school, I was really interested in biology and computer science, but I wasn't really sure I could combine the two into research. And that only really changed towards the beginning of sophomore year, which is around 2 years ago, when I listened to this talk by Professor Brendan Frey, who started a company called Deep Genomics. And Deep Genomics is the therapeutics company that's working on gene therapies for targeting rare genetic diseases with like less than 100,000 active cases. And the reason why this is a really important problem is with some of these orphan rare gene diseases, there's not enough of a sort of a incentive for larger companies to work on them because there's such a small sample size that would receive the therapeutic. And with that, there's a lot of R&D to get the therapeutic out into the market. And so what they did is they actually generated and screened the first AI-discovered gene therapy that's now going into clinical trials. So that sort of gave me the motivation to sort of think about how I could apply sort of my dual interests in computer science and biology together to try to solve this problem. And so the key point or thesis I kind of want to share with you guys today is sort of that we can shape a lot of these biological problems and chemical problems as computer science problems and machine learning problems. And this is largely because we have so much genome sequencing data now. It used to cost $100 million to sequence a genome in, say, the early 2000s. But now, with the help of a company called Illumina, they brought that cost down to $47 in 2020. So I think that's largely increasing the amount of open source genome data sets, the number of molecule data sets. And that was the biggest barrier for us to applying machine learning, because most of you know that machine learning just requires a lot of data to learn on, and we did not have that data 10 years ago. So we're sort of positioned in this unique time frame in sort of humanity now that we finally sort of have not only the research, but we also have the data to actually go forward and apply this to drug discovery. And so some of my earlier work before I found out about Emergent Ventures was basically trying to represent genome data as a vector and trying to feed these into deep neural networks for trying to understand genome biology. So most of you know about sort of computer vision and deep learning, and sort of that's the technology that actually powers autonomous vehicles and sort of object detection that's used in cars like the Teslas and a lot of those different technologies. But basically, what I was really interested in researching is, could we apply this similar architecture, but apply it to an image-based representation of the genome? So the genome is made of like 3 billion A's, C's, G's, and T's. And I wanted to see, can we represent this as a vector, just like we would represent an image, and then feed these into deep neural networks for predicting potential mutations that impact protein binding sites. And so through this research, I was able to basically look at one gene called the LDR1 gene, which is known as the cholesterol gene that causes higher cholesterol levels. And one of the things we found was that we basically were able to identify certain positive mutations that basically increase the likelihood of a protein binding to a DNA sequence. But we also found certain mutations, such as the C mutations, that tend to have a really high negative impact on DNA protein binding. And so this early work sort of was the sort of beginning of my journey into like machine learning and computational biology. But last summer, I took a little bit of I wanted to try learning about a different field, which was actually software and machine learning, mainly because I knew I could do research, but I wanted to say, could I take these models and actually deploy them in such a way that they actually reach users and actually are accessible to the world? And so through my internship at a company called Integrate AI, I actually focused on how can I take these different architectures like computer vision and all these architectures and actually apply them to different applied ML cases, like predicting turnover in customers and actually how to deploy these big complex machine learning algorithms. And after doing that, I think that was really the background I needed to start actually working on building open source drug discovery. So since then, I started working on DeepChem, which is this open source drug discovery project created at Stanford that basically is aiming to accelerate basically machine learning for the life sciences. So we've created tutorials which have thousands of different users dedicated towards applying machine learning for genomes, molecules, proteins, cellular images, and much more. And so far, the library has received over 2,400 stars and contributors across the world. And I think through the DeepChem project, I actually learned a lot about gaining the necessary experience about working and doing research in computational biology, chemistry, and machine learning, along with how to actually basically build scalable infrastructure. So through the developer calls, I was able to get mentorship from a lot of other researchers that were working on this project pro bono. And it actually gave me a lot of the knowledge that was, say, previously I would have had to go through academia or, say, an industry internship to get. So I think that really cites one of the biggest benefits of open source is it's a great place to learn. It's a really great place to provide mentorship and sort of bring and sort of inspire new researchers to sort of get involved in this, involved in this field of, say, computer science and chemistry. So as part of my research that was supported by the Emergent Ventures, I basically focused on developing these large-scale language models called transformers and the relevant utilities to actually use these language models on predicting different features about molecules, such as whether a molecule is toxic or not, whether that molecule will bind to a protein target, and various other drug properties. And so through this project, the transformer models were all open source. And one thing that I tried to do from the founding of this project was to actually build a research project from the get-go completely open source. So when you look at how most machine learning research projects are done, Typically, they'll build like silently. They won't share their work with anyone out of fear of the scooping. And then they'll release the paper and then release release their GitHub library. And sort of some of the issues with this is that a lot of the libraries don't end up being very reproducible. So you'll end up having many, many bugs with actually trying to replicate the results that, say, an author has shown in their benchmarks. And there's various other issues that basically are sort of contributing to this sort of research reproducibility crisis that a lot of people are talking about in computer science and the life sciences. So I think from the get-go, I wanted to not only build meaningful research, but actually build a project which people would use, and not just a project that would end up culminating in a nice publication, but then no one would ever use again after a year. So that was a really, really fun experience. Just to explain a little bit more about natural language processing. Basically, NLP is this sort of architecture that aims to look at language models. And these language models are popularly used in Google Translate, Siri, or even autocomplete. And you might have even heard of a company called OpenAI, which recently developed GPT-3. And GPT-3 is this architecture that's making a ton of waves throughout the natural language processing community and generally all of Silicon Valley, because it's this huge model that costs over $12 million to train, that can sort of generate text with just a simple prompt. And it can do anything from generating music to say writing React code just by fine-tuning it on, say, a smaller data set. And so that's something that's really sort of elevated the field of natural language processing. But what I wanted to do is I wanted to say, could we take this same architecture, the transformer model, and actually apply it towards understanding chemistry? So we've successfully figured out ways to, say, classify text, generate new text with a high degree of accuracy, but can we learn features about molecules by representing these molecules as strings, and then trying to get a model like this to predict toxicity? And so through this project, which was supported by U of T and also the DeepChem contributors, we basically were able to develop this architecture that as we pre-train and train it on larger and larger data sets, it becomes more and more accurate at predicting things such as toxicity. And I think the game changer with a lot of computational models is if you can get this transfer learning technology, which OpenAI has done, actually implemented, that's one of the biggest barriers for applying computational models to pharmaceuticals. So when you're working for a pharmaceutical company, you don't have these wonderful, really clean data sets with hundreds of thousands of samples that are evenly balanced when you're, say, trying to predict something. You'll have these really, really tiny data sets with maybe 1,000 or 2,000 molecules that are incredibly imbalanced. And so it makes it really hard for a machine learning model to learn on. And so I wanted to try to develop transfer learning in a way that we can pre-train it on a different task and then basically fine-tune or apply these models to one of these smaller data sets and still achieve a really high accuracy. And I think if we're able to keep developing this technology, which has become sort of one of the biggest problems in this field of machine learning and chemistry, that's one of the sort of breakthroughs we need to actually get machine learning and get computer science applied at scale in pharmaceuticals. And another really cool part of this is we were also able to try to visualize different parts of a molecule using this model. So with machine learning, another big problem with deep learning is just interpretability and trying to look at your weights in your model and figure out what is this model actually learning, because it's running millions of these different computations based out of, say, your AP calculus class that you are really, really hard to trace. And so basically I worked on building some JavaScript to actually take a look at how this model looks at every single token in a molecule and how it places weights on different tokens and on different substructures. And the goal with this is you can really understand, is this model learning a good representation of this molecule? Like, has it identified what are the most important parts of this molecule for, say, predicting toxicity or predicting its properties? And so in this diagram, we look at a graph-based way of representing this visualization. And so here you can see that darker highlights actually mean that this atom is much more important in the overall structure than, say, the lighter highlights. And I think what's really cool about these techniques, other than the fact that they produce these fancy looking diagrams, is they might sort of open up another level of understanding towards what our model is actually learning, if our model is actually learning something valuable, and also even towards chemists, hopefully one day in the future, actually looking at different parts of a molecule or substructures. We like to call them functional groups in chemistry that account for a large portion of the molecule's characteristics, which I think has a large application towards drug design. And another thing I really want to stress with this project was that this was entirely created through open source collaborators throughout industry and academia. So we never had any academic or corporate partnerships for example, cloud compute. So this is completely done by obviously the DeepChem project and the different contributors there with their help, and obviously with the mentorship of some of my mentor at U of T. But I think this really shows that hopefully in the future we can start to see more projects like this that are basically open source research that are done independently of, say, academia or some pharmaceutical company, just to sort of open the sort of different, more diverse basically backgrounds into this research field, and also bring more novel research from basically different places across the world. So now with this project, we're actually currently in discussions with different startups, companies, and different universities just for the cloud compute to do GPU training, because again, with a lot of these models, the GPU training is definitely not cheap. And so this project is entirely open source. So compute donations of any kind for AWS or GCP are definitely heavily appreciated. So kind of just to wrap up, sort of talking about open source, I also spent a little bit of time working on a project called SELFIES. And SELFIES is this basically molecular string representation that was one of the foundations for wanting, for my inspiration to work on language models. And so with this project, you're actually able to create 100% valid molecule representation using a string. So you don't need a really complex 3D conformation. And when you apply these strings towards these generative models, such as a GAN or a genetic algorithm, you're actually able to generate batches of molecules which are far more diverse. And why diversity is sort of important is because when you look at the total chemical space, there are actually 10 to the 64 different molecules that we can actually synthesize. And we've only really looked at a fraction of that in the process of drug discovery. So creating these sort of, basically working on research that creates more diverse molecules is a really, really important subfield. And also through this, I've gotten to talk to many different people who use our library for various different research across the world. And I think it really shows the power of open source. Because I think one thing that I definitely want to hit home is that a lot of these open source projects may not get the same coverage as a new paper in, say, Nature or another really big journal. But in reality, the impact that they have on the actual research field is a lot more, because they're powering a lot of the research that's being done, and they're helping facilitate basically new research methods. You know, libraries like DeepChem or TensorFlow, looking at TensorFlow or PyTorch, or a library like NumPy in machine learning. Those are used in almost every single machine learning paper. And so when you think about that has almost an industry-wide implication on like millions of people, and software that goes out to impact billions of people, really, if you think about it. And it's completely open source. So I think that's something that really sort of reminded me of the importance of open source. And another thing I also want to mention is that open source isn't just being used for research. It's actually now being used towards the fight for COVID. So there's this project called the COVID-moonshot. That's this huge international group of researchers from academia, from labs, from companies across the world that are actually trying to facilitate in lab screening and computational screening of tens of thousands of different antivirals. So they've synthesized thousands of molecules so far, and they've actually tested these on SARS-CoV-2 samples in Oxford. And all of the computational power is actually provided by this effort called Folding at Home. And Folding at Home is this global distributed computing initiative that basically allows anyone with a computer to actually donate some of their GPU compute and some of their CPU compute towards protein folding, or basically trying to understand the COVID-19 spike protein structure and test some of these molecules and simulate the process of these molecules binding to the protein, kind of like that image there. That's actually one example of a molecule actually interacting with one of the complexes inside the COVID-19 spike protein. And this is a computational screening that's being done on someone's GPU computer, actually. So I think that's actually another huge thing that's being supported by companies like NVIDIA, and actually PC gamers, which have come to the rescue and donated their GPU compute. So I think that's another really powerful example of how open source science is actually in the now and not in the 10 years from now. Yeah, so the last part, just touching on COVID, was that our lab also worked on a project towards COVID in silico testing. And basically what we wanted to do is we wanted to develop a set of algorithms which can screen over these molecules and predict how likely they are to bind to the COVID-19 protein, this spike protein that I mentioned. And so we started off with this initial hypothesis of representing molecules as graphs, and then moved on to actually running these on over 30,000 molecules and then testing them using computational and in vivo screening. So to kind of explain a little bit more about how small molecule drugs work, how they simply work is that they bind to these little pockets, like I showed in that diagram before, in a select protein. And doing this can actually break the protein and sort of break the process of replicating. And when you look at viruses, all they do is they need to replicate. So they have these proteases, like the 3CL protease, that are all they're doing is they're replicating, and they're coding for replicating and replicating and replicating, and they need to keep replicating to stay alive in the host body. And so if you can obviously design a molecule that sort of slows down the replication process or completely stops it, then the virus is gone after a couple days of taking a drug like this. So that's why people are looking at drugs like remdesivir and a lot of the other different molecules. And so the way I did this is I was using a graph-based machine learning method called geometric deep learning. And I was able to represent molecules as graphs and screen over 30,000 of these different molecules, all of which are publicly available FDA-cleared drugs. So the reason why we're not just generating new ones and using FDA-cleared drugs is because they're much more likely to be approved quickly if there is some magic bullet that actually does show a lot of signs. So that's part of the process of why remdesivir and lopinavir and a lot of these molecules by things like Gilead, different companies like you might have heard in the news or moving around in the stock market. The reason why they're in the lead is they already have drugs for other previous viruses or diseases like Ebola that are successful, somewhat successful, I would say, in targeting COVID. And the way we really do this is we're actually taking data from SARS-CoV-1, which is sort of the big brother or the older cousin of SARS-CoV-2, because we have a lot more data about how SARS-CoV-1's protein and how different molecules are able to bind to it. But we don't have a lot for SARS-CoV-2 just because of how recent this pandemic occurred in when we first started working on this project. And so by using data and training it from SARS-CoV-1, we then run predictions on SARS-CoV-2 molecules, and we have a high degree of accuracy when we transfer over. And so with 30,000 different molecules, we eventually get down to 10 different compounds, which are going into in vitro and computational screening with the help of folding at home. And again, this is just another example of how open source projects like folding at home, which anyone can contribute to, even anyone, if you have a GPU, you can help this project. And so this is a really, really important, I think, process that sort of shows the power of open source science once again. And yeah, that's just some of the molecules that were basically found to have a high binding affinity to the protein in SARS-CoV-2. So thanks for listening. I hope if you have any questions about machine learning, drug discovery, open source projects, DeepChem, really anything, I'd love to start a discussion going and answer any questions and sort of get an interesting discussion going. But thanks.
Thanks, Sion. That's super cool. I need to jump to another call, so I'm sorry that I can't stick around for the discussion. Looks like a really neat project. I mean, I'd love to hear a lot more, but I was about to drop a chat and say thanks, I got to run. But
no worries, thanks.
Kyle, Chitty, nice to meet you, or almost meet you, see you without actually. Yeah. Hope to see you guys around at another one of these sometime. See you later, guys.
Awesome. Cheers. Man, that was super interesting.